Hello my dear students, I am Dr. Vaishaly Bharambe, Professor Anatomy. Students, are you aware that there is one temporary midline structure in the body that decides where your brain will develop, where your spinal cord will develop, where your vertebral column will develop? Such an important structure. Yet, after doing all this, it simply disappears. This structure is the notochord, and that's what we're going to study today. In the third week of Intrauterine Development Lecture Part 2, we will be studying the notochord. Come. So here you're looking at the epiblast, the hypoblast. This is day 15. This is the primitive streak. By process of invagination, cells have started to move below the epiblast. You can see them moving to form intraembryonic mesoderm, the third layer. You can see them replacing the hypoblastic cells to form endodermic layer. But there are some cells which decide to travel the midline. Extending from the primitive pit to the precordial plate, these cells form what is called as the notochord. That's the topic for today. Come. So what is notochord? What is its extent? How is it formed? What are its functions, fate and remnants and applied aspects? That's what we're going to cover today in this short lecture. Come. So notochord is a midline rod of mesodermal cells that form the axis of the body. They result in induce the development of vertebral column and the nervous system. So very, very critical. Okay. Come. So what happens? It's a group of cells that extends from the cranial end of the primitive streak to the caudal end of the precaudal plate. Okay. Put that in your brain. Cranial end of primitive streak, caudal end of the precaudal plate. How is it formed? So what happens, if you recall, at the cranial end of the primitive streak, there was an elevation of cells, a knob of cells formed, which was called as the primitive node or the Hensen's node. Soon it developed a pit in the center called the primitive pit. Now what happens? Take a look. First orient yourself to this, then I'll talk. Okay. This is the embryo. These are epiblastic cells, hypoblastic layer. This is the amniotic cavity. This is the secondary yolk sac. This is the connecting stalk connecting this embryo to the placenta. Here you can see the epiblast and hypoblast connecting to each other tightly, precaudal plate. Similarly, more caudally, cloacal membrane. This is the embryo we have now. Let's take a section of it. This is what it's going to look like. Amniotic cavity, secondary yolk sac, and now there is a new layer formed here, the intraembryonic mesoderm, by process of gastrulation. Right. Now what happens? Cells begin to depress downwards through the primitive pit, traveling towards the precaudal plate. I want you to use this image to really build a beautiful mind picture. See. This is the primitive pit. This is the notochordal process. That is the group of cells that have started to walk in the midline towards the precaudal plate. A midline cord of cells called as the notochordal process. From where to where? Primitive pit down, forwards to the posterior end of the precaudal plate. It's a cord of cells, collection of cells. Notochordal process. Take a look at a section of it over here. This in the center is the notochordal process. Soon the notochordal process develops a canal in it because the depression of the pit extends inside it. So now from a notochordal process, it gets converted into a notochordal canal. Okay. So it's like a tube-like extension of the primitive pit itself. Okay. See, here is your notochordal canal. Now something very funny happens. The floor of the notochordal canal begins to get intercalated with the roof of the secondary yolk sac. See here. Okay, so now the cells are mixing and suddenly the canal is communicating with the secondary yolk sac. If you are enjoying this lecture, do please like, share and subscribe to my channel and hit that bell icon. And do visit my website vbanatomy.com. Link is in the description section. Let's come back. I think we were having a great time. So see how there is a communication of the notochordal cavity and the secondary yolk sac because the cells have got intercalated as seen here. Okay. So this is the notochordal canal and it's communicating clearly with the secondary yolk sac. So roof of secondary yolk sac is now partly formed by the notochordal canal. Do you agree with me? Yes? I'm glad. So now that is the notochordal canal, but more than that, suddenly there is a communication between the secondary yolk sac and the amniotic cavity. This communication is called the neuroenteric canal. It's a big event. Why did it happen? Why did the secondary yolk sac and amniotic cavity, who never communicated till today, suddenly develop a canal in between them? I'll tell you why. Because what is happening is that lot of activity is happening in the epiblast, whereas the amniotic cavity does not have enough nourishment at this stage. So what the body does is a temporary, help is organized from secondary yolk sac. Nourishment comes from secondary yolk sac through the neuroenteric canal to the epiblast to help the cells obtain nourishment till the placenta begins to function properly. So that is the reason for nourishment is the reason why formation of a neuroenteric canal takes place. Meanwhile, placenta is developing. Okay. The canal now slowly begins to separate from the yolk sac again. Okay. You can see slowly the roof has started to form again. Definitive endoderm is being formed. So, neuroenteric canal is slowly reducing in size and the roof of the notochord now becomes thickened to form a notochordal plate. I'll show you. See, take a look at it here. The definitive endoderm has got formed. The notochord has become thickened, notochordal canal has become thickened to form notochordal plate, and the endodermal layer has started to slowly get completed again. See here. See how the secondary yolk sac roof is again complete. Alright. So again the plate bends, folds and finally forms a tube. This tube now is called as the notochordal tube. Okay, neuroenteric canal disappears, endodermal layer is complete. The roof is again formed by endodermal layer, but the notochordal plate, which was thickened, bent to form a tube. So you have a notochordal tube. Soon the notochordal tube gets packed up with cells, and what is formed is called as the notochord. How do you differentiate between the first and the last section? You can see that the intraembryonic mesoderm has also started to change into 3 longitudinal columns of cells. You can see the paraxial mesoderm, intermediate mesoderm and the lateral plate mesoderm. And this in the center is your notochord, the definitive notochord. So let's revise by drawing. Okay. Please draw with me, dear students. Let's draw the amniotic cavity, precordial plate, epiblast, cloacal membrane, hypoblast, allantois, yolk sac. Let's label them. This small little dot that you are seeing is the Hensen's node or the primitive pit. Now, how do we form a notochord out of this? What diagram should we draw? Come, we will focus only on the trilaminar embryo bit. Okay, so let's draw the amniotic cavity again. The cloacal membrane, precordial plate, amniotic cavity, hypoblast, allantois. So we are seeing the upper part of yolk sac and the lower part of amniotic cavity. That should be clear to you. Now you will see how cells begin to grow forwards from the primitive pit to form a solid cord of cells, which is the notochordal process. This is step 1 in the formation of notochord. Let's draw the next diagram now. Again, amniotic cavity, lower part. But this time, as you can see, a cavity has extended inside the notochord. Okay, so that from a notochordal process, it has become into a notochordal canal. Let's draw the hypoblast now. So this is your diagram number 2, formation of notochordal canal. In the next diagram, you're going to draw the canal in such a way that its lower surface is adjacent to the hypoblastic cells and their cells are undergoing intercalation. Come, let's draw that. So again, amniotic cavity, precordial plate, a very wide notochordal canal, cloacal membrane, yolk sac. See how closely I am getting the hypoblast cells to run along the notochord. Thus, the roof of yolk sac and the floor of notochordal canal are almost adjacent to each other. Now see how you rub out bits to show that the cells have undergone intercalation. Now, can you understand that part of the roof of yolk sac is now formed by the notochordal canal itself? Let's go to diagram number 4, where there is clear formation of a neuroenteric canal. That means a clear communication between the yolk sac and the amniotic cavity. So can you see how we are slowly developing a tubular connection between the yolk sac and the amniotic cavity? See, draw this line to indicate that this is the neuroenteric canal communicating the amniotic cavity above with the yolk sac below. This is your diagram number 4. Now, as the roof of this neuroenteric canal gets thickened, it forms a notochordal plate, which is the diagram number 5, formation of notochordal plate. Now what happens? How does this get converted to a notochord? Let's draw the notochordal plate, but it has now formed a notochordal tube. It has moved away from the hypoblast. The hypoblast, as you can see, is complete again and again forms the roof of the yolk sac. This is your diagram number 6, and finally you will see formation of a definitive notochord when this notochordal tube gets filled up with cells to form a solid cord of cells called the definitive notochord. As you can see, the yolk sac is again having the hypoblast as a roof. This is diagram number 7, completion of formation of notochord. Now all you have to do is color up the various layers to make it, give it an appearance of, neat and tidiness since you're drawing this for your exam. So color the hypoblast yellow, color your amniotic cavity maybe pink or any other color. There, that completes your diagram of notochord formation. Come, let's discuss some more. So what is the function of notochord? Notochord establishes the longitudinal axis of the embryo. It's a primary inducing development of neural plate and therefore neural tube and the central nervous system. It also guides the development of vertebral column. It persists as a nucleus pulposus in the intervertebral disc. With this, we come to the next common heading. What is the fate and the remnant of notochord? Notochord will of course disappear, but it will persist in the body as nucleus pulposus and apical ligament of dens, which are remnants of notochord. Just remember that a lot of books say that even these degenerate by age of 10. Sometimes notochord can persist, resulting in benign and malignant tumors called chordomas. These are usually found at the base of cranium, sometimes in the nasopharynx as well. So what did we discuss today? In this short and very sweet lecture, we discussed what is notochord, what are the steps in the formation of notochord, what is the fate and remnant of notochord, what function does it perform, and finally, its applied aspects. This lecture should ideally be useful for you to attempt a very common short note called notochord. So write a note on notochord. I've attached a PDF to this in my video. Do please download and use that for revision. So that was the end of third week of intrauterine development of the embryo. We covered a gastrulation lecture earlier, and today we've covered the notochord. I'm going to carry us now into the fourth week where a lot of activities are happening. Do come with me into the fourth week of embryo's life. Link to these two lectures is given in the description section. We also have a whole lot of general embryology other lectures. Okay, do please watch and feel free to ask me doubts. I'll be more than happy to clarify them. If you've enjoyed this lecture, do please like, share and subscribe to my channel and hit that bell icon. Visit my website vbanatomy.com for lectures, for surgical anatomy videos, for downloadable notes, MCQs. Okay, link is in the description section. I'm on a journey with the little embryo. I'm going to be with the embryo, allowing it to grow, helping it to grow, nudging it to grow from third week into the fourth week. Come. I'll see you in my next lecture.